Well, hello, and welcome to day 2 of the Sustainable Protein Forum Student ePoster Pitch Competition from AOCS, which is sponsored by Kelsic and the Good Food Institute. My name is Dr. Jim House, and I'll be moderating today's session. I currently serve as a professor in the Department of Food and Human Nutritional Sciences at the University of Manitoba in Winnipeg, Manitoba, Canada. We wish the students well and thank all of our volunteers and our volunteer judges and the audience for attending, listening, and supporting this competition. Yesterday, we had an excellent event with four presenters, and we are looking forward to 3 presentations today. But to start off, I'd like to introduce our judges. Our judges today will be Carrie Campbell, who is Process R&D Engineer for Roquette Americas, Dr. Elaine Cruel, who is President of EKSI LLC in the US, Dr. Chris Marinangeli, a Director of the Regulatory Center of Excellence for Protein Industries Canada, and Mahfuza Rahman, who's a Senior Scientist of R&D for Kraft Heinz Food Company in the US. Thank you, judges, for dedicating your time and expertise to this really important exercise. But let's get to what will happen today. Today, we will be hearing from 3 of the competition finalists, and each finalist will present for up to 5 minutes. They may use a visual aid like a PowerPoint, but not their actual ePoster. If you would like to view the entire ePoster, please visit the Sustainable Protein Forum website. After each student presents, a 7-minute question and feedback session will take place. And we encourage the audience to post any questions, comments, or even congratulatory notes in the chat area. And I'll be forwarding those on to the actual presenters as we progress through the question and answer session of today's event. So please use the chat feature to introduce your questions as we proceed, because we will have time after each presenter, as well as at the end, we will have a final wrap-up opportunity to ask additional questions. After all 3 presentations have ended, as I mentioned, we'll bring all the finalists and judges back on screen for any remaining questions and feedback. And at that time, we'll open up an audience poll so that you, our live audience, can vote for your favorite pitch. So it's a really important exercise because that will actually be included in the final score as we evaluate the final 7 presenters for their presentations. So please keep in mind that in order to participate in the voting, attendees must access the live stream on the Sustainable Protein Forum website. So I encourage you to do that. The announcement of the winners will actually take place next week during the Sustainable Protein Forum, which is happening in Chicago. So it's in both a virtual and an in-person event. And we encourage you to check the website for more details on the forum if you are interested in attending. But why don't we get started by introducing our first presenter. So our first presenter is Peineng Zhu, who is a PhD candidate at the University of Alberta in Canada. Peineng received their bachelor's degree in food science and technology with a specialization within those disciplines, and her research focuses on tailoring pea protein gelation for novel and conventional applications in food and biomaterial fields. Peineng will present today on preparation of transparent and thermo-reversible pea protein gels and their potential applications as gelatin alternatives. Good luck, Peineng. And we will pause while we prepare for the presentation.
Good morning, everyone. My name is Peineng, and thank you all for attending my poster presentation, which is about develop and characterize thermally reversible transparent gels from pea protein as a potential alternative for gelatin. As you may know, gelatin is one of the most popular biopolymers, and it has numerous applications as gelling agent in food, confectionery, cosmetic, pharmaceutical, and biomedical products. And gelatin are mainly produced from bovine and porcine sources, but gelatin may also be extracted from fish and poultry. And it shows thermo-reversible gelation behavior, where the protein transition between random coil and the triple helix through hydrogen bonds plays a key role to its thermo-reversibility. And in addition, the formation of transparent gel is also desired in many applications. However, more and more consumers are turning for plant protein sources and food products due to religious and health concerns. So gelatin alternatives from plant sources have gained increased interest. And many polysaccharides have been proposed for the food industry as gelatin alternatives such as starch, pectin, carrageenan, and many others. But gelatin alternative from plant protein source has not been reported before. So here we report a transparent thermo-reversible gel made from pea protein. And pea is a rich source of protein containing up to 30% protein with the advantages of high nutritional values, lower allergenicity, and free of genetic modification issue. So here is a brief flowchart telling how to prepare the gels. First, we extract the pea protein isolated from pea flour through salt precipitation method. Then by preparing the pea protein solution at a relatively acidic pH, where it is highly soluble. Afterwards, the pea protein solution was heated for only 10 minutes and followed by cooling for the gel formation. And conditions in terms of pH, protein concentration, melting, and the gelling temperature for the thermo-reversible gelation have been identified. And the mechanism of gel formation was also studied through HPLC analysis to know the protein profile and the gel dissociation test to understand the involved molecular interactions in the gel. So we look at the result. You can see that through 10 cycles of heating and cooling process, the thermo-reversibility and appearance of pea protein gels was consistent. And also from the dynamic rheological measurement within 2 cycles of heating and cooling process here, it also disclosed the thermo-reversibility of the gel being stable, while an instant drop of G prime value is observed upon the temperature increase, while a rapid increase in the G prime value during cooling corresponded to the protein gelation. And through the analysis of protein profile and the gel dissociation test, which is not shown here, but in the poster, we propose a gel formation mechanism where pea protein molecules were dissociated to smaller subunits or monomers at acidic pH and formed the polymer-like structure, which were physically cross-linked through hydrogen bonds that can be disrupted at higher temperature and reformed during the cooling process. Since this pea protein gel shows similar characteristics of being transparent and thermo-reversible as that of gelatin gel, which makes it possible as a potential alternative to replace gelatin in many food formulations and in cosmetic applications as gelling or thickening agent to provide clarity and texture. And in other non-food area, it might also be possible to be used for the preparation of transparent capsules or serve as carriers for nutraceuticals in delivery system. Yeah, that's all for my presentation. Thank you for your attention.
Thank you, Peineng. Your presentation was excellent, very clear. And now it's time to turn it over to the judges who will be asking some questions and providing some feedback. Just a reminder for those in our audience, please add your questions and comments in the chat box, and we will share those with the presenter and ask those during the feedback session. So why don't we start this time? I'll cycle through the judges in no particular order, but let me select Dr. Chris Marinangeli first to ask the first question of Peineng. Chris?
Thanks, Jim. Excellent presentation. Thank you for that. The question that I'm going to ask and focus on is the claim on one of your first slides around nutritional quality. I'm just wondering how you're characterizing nutritional quality compared to traditional gelatins in the marketplace.
Okay, we analyze the amino acid profiles and also the PDCAS, which is the amino acid protein profile for digestions. But like in these studies, we didn't focus on the nutritional value of the pea protein, so we focus on the functionalities of the pea protein compared to the gelatin.
Did you come up with a? Do you have a PDCAS for the gels?
For the gels, no, I don't have that.
Okay, and just I guess a follow-up question along similar lines regarding the claim around the hypoallergenicity. I'm just wondering if there's former experimentation that have been done for that claim made in your presentation.
So for the there's not many studies like focusing on the allergenicity studies of pea protein. But compared to the soy protein, I think I remember there's a study like comparing like and so the conclusion will be the pea protein has a lower allergenicity compared to soy protein.
Great. I'll pass the baton, Jim.
Okay, thanks, Chris. Why don't we ask Mafazer if he wanted to go next in the question? Thank you.
Thank you. Very good presentation for sure. So during the preparation of pea protein isolate, you have used the salt precipitation method, which is not common. The conventional method is alkali and like isoelectric precipitation. Like, why did you choose this method, and like what happened if you extract using the conventional method?
Yeah, I chose salt to precipitation method. Like it has less damage to the pea protein structure compared to the conventional alkaline extraction method. But as you may say, there's advantage and disadvantage. So the disadvantage will be it has a lower yield compared to the conventional method, but the protection of the protein profile will be better if we use the salt precipitation method.
Have you compared your gel with a alkali extracted protein isolate?
Yes, we compared that, but only for this salt precipitation method, we get a gel with thermo-reversibility and the transparent appearance, but not the conventional extraction method.
Great. That is an important point you should mention. Thank you so much.
Yes.
I'll pass to Dr. House.
Okay, thank you, Mafazer. Elaine, would you like to ask next?
Yeah, that was a wonderful presentation. Peineng, have you tried, is this unique to pea protein, or have you tried any other plant-based proteins in your process to see whether or not you can get something similar?
I would say it's unique for the pea protein, but I also know that the farbabin has a similar protein profile, and I think it's very possible if we extract the farbabin like in the same method, the protein can also have the functionality of having a transparent and thermo-reversible pea protein gel.
Thank you.
Great, thanks, Elaine. And Carrie, would you like to ask a question now?
Yeah, I was wondering what was the purity of your protein isolate, and how would how, could you comment on how some impurities may affect your gelation properties?
Yeah, I tested the purity. It's about 95%.
Okay.
So it's a pretty high purity of the pea protein.
Did you do any characterization of what that other 5% may be?
Oh, it should be, but I didn't characterize it, but usually it should be like fibers, polyphenols.
Okay. Yeah, I was just wondering because the isoelectric precipitation usually is more difficult to get a high purity like that. So I was wondering if that may be another explanation why the isoelectric precipitation didn't work as opposed to just denaturation, because the heat and acid pH that's pretty strong denaturing conditions as it is. It was I was just wondering.
Yeah, that's one point.
Okay. All right, thanks.
Thank you.
Okay, thank you, Carrie. We do have some questions from the audience, and so I'll pose those now. One question was this. I guess this is a question for you in terms of just what your thinking is relative to the balance between nutritional properties and functional properties. Where do you put the emphasis when you're designing a gel product, such as what you've done during your exercise?
Yeah, when we are doing the exploring the functionalities of pea protein in terms of its gelling properties, we focus more on the functionalities. So in this case, we didn't really measure its nutritional profiles. But since the method and the extraction method we use is it's food grade, so the salt precipitation method should protect the protein profiles. So in this case, we think it get the both way, like it protect the nutritional profile as well as it gets a good functionalities. So we will concern about the extraction and the method process that we get the protein, and also we care much about what functionality it can get.
Okay, great. We have other questions coming in, but I think we'll save those for the final round. So thank you very much, Peineng. We'll take a short break here as we prepare for the next presenter. So thank you very much.
Thank you very much.
Welcome back. Our second finalist, Yuran Zhang, is a graduate student at the University of British Columbia in Vancouver, Canada. Yuran obtained her bachelor's degree in food science and technology at the Zhang University. And Yuran is currently working on the fibril core regions of legume seed storage protein fibrils in Dr. Derek Dee's laboratory. Yuran will be presenting today on identifying the core regions of legume seed storage protein amyloid fibrils. So good luck, Yuran, and we invite you to present to the group right now.
Thank you so much. Hi everyone, my name is Yuran from the University of British Columbia. Today it's my great pleasure to present my research, which is titled Identifying the Core Regions of Legume Seed Storage Protein Amyloid Fibrils. When you see the sentence from diseases to sustainable functional ingredients here, you might wonder what does it do with diseases? Well, if you're familiar with Alzheimer's or Parkinson's diseases, you might know that amyloid fibrils has been long considered as one of the main causes of many neurodegenerative diseases. However, nowadays, more researchers have discovered that many amyloid fibrils, especially those derived from foods, actually display excellent functionalities such as gelling, emulsifying, foaming capacities that could be used in various applications such as fat substitutes. In this field of research, most existing studies, they use dairy proteins for making food protein amyloid fibrils. However, fibrillation is actually generic among a wide range of proteins, including legume seed storage protein, which mainly consists of 7S and 11S globulins. So why do we care about legume proteins specifically? Well, first of all, legume proteins require less water and land resources in production than many traditional animal-based proteins, and it has lower greenhouse gas emissions. Moreover, consumers nowadays, more and more consumers are willing to accept legume-based proteins as substitutes for traditional animal-based proteins in today's food markets owing to rising health and sustainability awareness. So how does fibrillation occur in vitro? Well, it usually happens under high temperature and low pH conditions where the native protein will further undergo hydrolysis into peptides, and some of its peptides could be converted into a beta-sheet-rich protein aggregate, which is termed amyloid fibrils. In this fibril, there is a fibril core structure, which is essential to us because it is important for us to understand the mechanism of fibrillation, and also the fibril core itself could either accelerate, inhibit fibrillation, and even alter the fibril morphology. So based on these, we developed 3 research questions. First of all, how do fibrils from different legume globulins differ? Here we chose pea and soy as our models. Second of all, how do fibrils from different globulins differ? Here, the major globulins of C-surge protein will be 7S and 11S globulins. And lastly, we want to know how does the fibril core composition look like in this C-surge protein amyloid fibrils. So to answer these questions, we extracted crude extra 7S and 11S globulins from pea and soy using alkali extraction, and isolate 7S and 11S globulins using the combination of salt, pH, and FPLC. And then we incubate these proteins under pH 2, 80 degrees Celsius, and examine their fibrillation kinetics, morphology, and fibril core regions. So here's what we found. First of all, in terms of kinetics, we found that 7S globulins, either from pea or soy, display no lag phase, which means that they form fibrils faster than the 11S globulins and crude extracts. When we observe these fibrils under transmission electron microscopy, we see that pea and soy globulins form very different fibrils, where the pea fibrils are straight and soy fibrils are rather worm-like. What we also saw is that the extended incubation from 30 to 78 hours seemed to disrupt the fibril network that has been formed by 30 hours of incubation. Finally, in terms of the fibril core structure, we see that P7S globulin, among all the samples, possesses the most abundant fibril-forming regions, which is around over 70% of its entire sequence, which is surprising to us. And when we compare our results with the previously studied ovalbumin and beta-lactoglobulin, we saw that most of our legume globulins are richer in fibril-forming regions than these dairy proteins. In summary, we found that pea and soy 7S and 11S globulins all exhibit strong capacity of forming fibrils with distinct morphology and kinetics. And they all show potential of replacing dairy proteins for making food protein amyloids. So with this knowledge, we hope to build a groundwork for tailoring legume proteins for board and structure and functions, and understand the mechanism of legume protein fibrillation. And with that, ultimately, we want to change the reputation of amyloid fibrils from the bad guy into the good guy. Thank you so much for your attention. I'll be happy to answer any questions you might have.
Thank you very much for that excellent presentation, Yuran. It was very informative. And now it's time to turn it over to our judges who will ask you some questions. And so I'd like to first start off by asking Elaine if she would start questioning for this next round.
Sure. I really enjoyed that talk. Yuran,
Thank you.
have you looked at the effects on digestibility of the proteins once they form fibrils? Even if it's an in vitro static test of digestibility, to see how that impacts the protein quality?
Yes, so we haven't done that experiments, but we have found some similar studies that have studied the digestibility of some animal-based food proteins, for example, beta-lactoglobulin. And what they found is that depends on the protein itself, they exhibit very different digestibility because some of the proteins, they're really prone to, let's say, enzyme digestion, or even just by putting them under extended incubation would disrupt the fibril network. But ultimately, they or exhibit some sort of resistance to enzyme digestion because of the unique structure of the fibril itself.
Mhm.
Thank you for that question.
Thank you.
Great. Thanks, Elaine. Carrie, would you like to ask the next question?
Sure. Yeah. Thanks, Yuran. And one of the big, for me, the big interest in fibril and proteins that create fibrils is in meat analogs, wet extrusion. And
Yes.
I was wondering how would you utilize your learnings here to improve extrusion processing for creating meat analogs with plant-based protein?
Yeah, thank you. That was a very interesting question. So I understand that in today's meat analog production, one of the main focus is to produce the fibrillar protein network. However, for many globulin-like protein, it is really difficult because they do not have the fibrillar structure in the first place. So I understand that they have many strategy like bottom-up or top-down strategy, where the top-down strategy may be like you mentioned the extrusion or some similar mechanical energy you put inside a system. But for this one, I think it's very unique because you form the fibrillar network for to use a really bottom-up strategy. So like you assemble these proteins in the fibrillar network in the first place, that you may require less mechanical energy input to shape their internal structure. So I think this might be a really unique perspective to adopt in the future development of meat analogs.
Okay, thank you.
Thank you for that question.
Great. Thanks for that. We'll ask Chris now if you'd like to ask a question at this stage.
Thank you, and to echo fellow judges, excellent presentation. My question's around just a comparison between the plant proteins that you compared in your work. I'm just wondering, based on the structure, do you see advantages for different food applications depending on the source material being used?
Yes, so thank you for that question. So in this research, our focus would be identify the fibril core. So we didn't really look into the functionality-wise applications, but we have seen in previous studies, so P proteins, they haven't separated 7S and 11S and studied their functionality separately. I mean, for like amyloid fibril format. But there has been studies that show that some proteins with different morphology, for example, some are short rod-like and some are really long and straight, and they will exhibit very different functionalities in terms of like viscosity. So I've read some papers, they show that the long and straight fibrils, depending on the sources, they will show like better thickening effect for the entire food system. But it really depends on the protein source, as you mentioned. So that's something we will look into in our future studies. Thank you.
Thank you.
Great, thanks, Chris. And Mafazur, would you like to ask a question at this time?
Thank you. This is a great presentation for sure. So I'm just curious to learn, like when you are hydrolyzing the protein, acid hydrolysis, are you like losing the functional properties of the protein? Like if we think from food formulation, whether it is meat analog or dairy analog, are you losing the functional properties or you are achieving better functional properties?
That's a good question. So if I'm understanding correctly, you are saying that the hydrolysis itself, does it cause inferior functionality or better functionality?
Yes.
Okay, so that really is a good question, but if I can remind you the process of fibrillation in vitro under high temperature and low pH, the process is you undergo hydrolysis, and some of its peptides could be converted into amyloid fibrils. So there are a lot of studies that study the conversion rate, which is not very high, around 50%. So it means that the entire system consists of some peptides and also some amyloid fibrils. So it's a mixture. So some studies they will investigate the fibril, the functionality of the fibril itself, or as a system. So one of the main focus in this field of study is to increase the conversion rate. But you're right, when you hydrolyze into peptides, it really depends on what kind of functionality that you're interested in. So there are studies that focus on the foaming capacity, and they found that the peptides itself in the system actually contribute to most of its foaming capacities instead of the fibrils. However, in some other functionalities such as viscosity, thickening effect, the fibril itself plays a better role than the peptides in the system. So it really depends on what kind of functionalities you're interested in.
So what is the reason behind the differences you just explained before?
Yes.
Foaming versus viscosity. Why so?
So I'm gonna start with the thickening and viscosity effect, just because that's easier. So the fibril itself, because of its efficiency of forming a really fibrillar strong network with a low protein composition, so it forms the elastic network easily. So it kind of increases the viscosity of the entire system. But in terms of like foaming, there are various explanations in this field of research. So there are some people explain that, because the peptides itself can be absorbed to the interface faster than the amyloid fibrils, and some explain that the fibrils itself do not have the high efficiency of absorbing to the interface, so they have worse stabilizing effect of the foaming capacity. But if you use another source of legume, some people argue that because amyloid fibrils could thicken this entire system, so the foaming capacity could be maintained at some level, but not as good as the peptide alone.
Okay, thank you.
Great, great. Thank you very much for answering those questions and for your excellent presentation. I think we'll end the questioning period for you at this point in time. And just a reminder to our audience attendees, please post your questions in the chat room, and we will answer them either during the presenter session or at the session that immediately follows the third presenter. So at this point in time, we'll take a quick break as we prepare for our third presenter. So thank you, Yuran.
Thank you.
And welcome back. And our third finalist and final finalist for today's session is Jessica Hallstrom, who is a PhD candidate at the University of California, Davis in the US. Jessica is a former senior food scientist in research and development at a manufacturer of vegan products. And her background in the food industry includes product development and commercialization from formulation to scale up. She plans to return to industry after completing her doctorate and to one day teach product development to the next generation of food science professionals. A very noble endeavor. So thank you very much, Jessica, for being here. And we welcome you now to present your talk entitled Impact of Roasting and Particle Size on Proteins Extracted Using Aqueous and Enzyme-Assisted Methods for Almond Milk Production. The floor is yours, Jessica.
Thank you, Dr. House, for the introduction. Hello, everyone. Today, I'm going to be discussing my research and using a green technology to produce a nutritionally improved almond milk and the impacts that degree of roast and particle size have on the proteins extracted using this method. There's a good chance that most, if not all of us on this call today have almond milk in our refrigerator. Almond milk is the most popular dairy alternative in the United States, but it is nutritionally inferior to bovine milk. Bovine milk has around 3.25% protein and 3.5% fat, whereas most commercial almond milk has less than 0.5% protein and only 1.25% lipids. This is a bit counterintuitive because almonds are nutritionally dense. but it's due to the manufacturing process used in most almond milk. A fine almond paste is suspended in a very dilute solution, along with gums and emulsifiers, at a ratio of around 1 to 50 almonds to water. Because there is so little actual almond in the milk, it is inherently low in nutrients. Our proposed solution to this problem is the use of Aqueous, or Enzyme-Assisted Aqueous Extraction Processing, or AEP/EAEP. This technology relies on the disruption of the almond material to expose its intracellular proteins, which are solubilized in an aqueous medium with agitation, increased temperature, adjusted pH, and time. When the protein is extracted, it leaves pores in the cells, leaving oil free to exit as well. The slurry is then centrifuged, and the lipid and protein-rich supernatant, or milk, is separated from the fiber-rich insolubles. We can enhance extraction with the use of an enzyme, in this case an alkaline endoprotease, to further improve total protein extraction as well as alter protein functional and physicochemical properties. Upstream processing in the form of degree of roast and size reduction, or grind, can also be utilized to tailor the properties of the almond milk. For this study, blanched deskinned almonds were either left unroasted, exposed to a light roast, or dark roast, and then ground to either flour, butter, or paste, yielding 9 raw materials. Following extraction, the milks were then analyzed for their protein, fat, moisture, ash, and total carbohydrate content. The milk was then defatted by a separation of cream from the skim, and skim milk proteins analyzed for surface hydrophobicity, setup potential, and secondary structure to assess the impact of roast and extraction method on these proteins. Roast was found to increase protein extractability from flour, likely due to the decrease in size in light and dark roasted flour compared to unroasted. This is to be expected as roasting facilitates grinding in nuts. However, in the finer grinds where particle size was more equal between roasts, roast actually impeded protein extraction, likely due to protein denaturation. Enzyme use increased extraction overall in the EAEP milks, with total protein extraction nearing 100% in all roasts and grinds that were finer than flour. Particle size had the largest impact on extraction in EAEP milks. And since extraction levels differed between butter and paste in all extractions, butter was chosen as a material to analyze milks from for deeper protein investigations. Circular dichrosum revealed that enzyme use in EAEP reduced alpha helix secondary structure percentage pretty significantly compared to AEP, as well as an increase in beta sheet structures. Roasting didn't greatly impact the structure of AEP extracted proteins, but darker roasting did lead to decreased beta turns and increased percentage of unordered protein structures in EAEP. And these circular dichrosum results may explain some of the functional properties we investigated. Overall hydrophobicity decreased as roast intensity increased in AEP extracted milks, indicating that the aggregation and denaturation of proteins induced by roasting might have hid hydrophobic sites. Surface hydrophobicity of unroasted and light roasted EAEP milk proteins were lower than those of all AEP milks, though statistically similar to that of the AEP dark roasted milk. The use of the enzyme likely cleaved and exposed sites that otherwise would be hidden by roasting AEP that may have led to increased ionization potential and thus increased repulsive forces. Interestingly, the dark roasted EAP milk was slightly higher, which may correlate with the increase in unordered structures found in this milk. The mean surface charge of all EAP skim milk proteins were significantly lower than those of AEP milks regardless of roast, though all are highly negative, indicating a strong electrostatic repulsive force. Ultimately, light roasted butter produced an almond milk that yielded a more than 300% increase in protein in EAP compared to commercial almond milk, indicating that almond milk with tailored nutritional and functional properties can be produced by AEP and EAP, as well as upstream processing modifications. This informed our future experiments, as well as including sensory analysis and scale-up, which are actually currently underway. And thank you for your time, and I'm happy to answer any questions about this research.
Excellent presentation. We'll now turn it over to our judges to ask you some questions. And I'd like to first call upon Mapazur, if you'd like to ask the first question.
Okay, thank you so much, Dr. House. Thank you, Jessica, for nice presentation. So yeah, if I understand correctly, you have not done any defatting and you have like good amount of fat and your end product as you have shown. So I'm very curious to learn about the effect of roasting on a undefatted flour, because like if we have an undefatted flour, it's already been very hexagonal type of flavor, and you already roasted those undefatted flour. Like, have you seen or have you tested those sensory or flavor analysis?
So what we actually did is I roasted the almonds whole, and then so there were three different levels of roast: unroasted, light roast, and dark roast. And then those were ground. So I didn't roast the flour and roast the whole almonds. And they were initially ground to a flour consistency. And the thing is, once you grind roasted nuts, it's not really flour anymore. It's kind of chunky almond butter, because the process of roasting shrivels cell walls, it bursts oil bodies, which releases free oil into the medium. That's why you don't see unroasted almond butter, or you don't see roasted almond flour, because once you roast it, there's so much free oil release that it sort of becomes like chunky almond butter. But the nice thing about that is that once you roast it, is that you get all those lovely nutty flavors. Unroasted almonds taste very different from roasted almonds. You've got those furans and the nice color changes. And we actually found, doing some taste testing, that unroasted almond milk has a very different flavor, that kind of raw benzaldehyde taste, versus a little bit of roast is really necessary to improve the flavor. And of course, there's going to be some changes to the oil quality with roasting, but we did not investigate that as part of this study.
Yeah, so even look, I have like serious doubt about the like oil quality or the like effect of oxidation on roasting, like effect of roasting on the oxidation of oil. Like we are struggling every day about this roasting or like the defatting, fatting on the flavor profile of our proteins we are using. So and another important thing here is like when you have full fat something, extracting protein is very hard, right?
Right.
And when you store for a long time, it's get oxidized over the time during the storage, And like when we get get those protein for the food formulation after 3 months, it really become like very hexagonal type of flavor. So therefore, I would recommend you to test do some flavor analysis, and if possible, do some storage analysis at the same time.
That's a potential. That's definitely a potential avenue for us. What the actual sensory testing that we're starting is going to be, so we have selected our material, the light roasted almond butter, or actually paste is what we're using for the scaled up sensory test. But the main things that we're evaluating in the sensory test are solid to liquid ratio, so quantity of almond paste used in the extraction, as well as extraction time and enzyme percentage. Storage wise, that's something that we're probably going to be looking at later when I do the full scale or pilot scale experiments, where I scale up using a UHT homogenizing and pasteurizing system, because the way I make it in the lab is kind of different from how it would be made in an actual manufacturing facility, where you're doing the high temperature short time or ultra high temperature pasteurization. And so that would, that very short time of pasteurization would help protect the oils quite a bit better than the current batch pasteurization method that I have to use in the lab right now.
Okay,
Great,
thank
great.
you. Thank you.
Thank you very much, Officer. Chris, can I ask you to pose a question?
Yeah, again, a great presentation. My question is around one of the statements in your initial slides around green technology, which I think is really interesting, considering all of the conversation happening around the world, the food sector around environmental sustainability. I don't think almonds have been able to steer clear of this story. So I'm just wondering, with some of the proposed, I guess, processing methodologies that you're using to generate your results, I'm just wondering if you've evaluated or looked at or hypothesized any effects around the sustainability profile if some of these technologies that you're bringing forward were scaled up into almond milk production.
It's definitely a tricky question in terms of almonds. I mean, we are in the Sacramento area where almonds are king, and they do, of course, require quite a bit of water. Of course, compared to dairy milk, the water consumption is much, much lower. And aqueous extraction also requires quite a bit of water. We consider it a green technology because we're extracting quite a bit of protein and oil without the use of hexanes, you know, and using any sort of more toxic inflammable solvents. So by comparison to that, it is considered a much more, you know, eco-friendly, novel technology. And ultimately, the goal of this is to create a more nutritionally dense almond milk that still has that creamy, rich flavor. Because something that I didn't get to mention in the first slides, the reason that almond paste is kept so low in most conventional almond milk is because if they added more of it, it would precipitate out of solution because it's got all that fiber. So our technology removes that fiber, or at least most of it, as much as possible, so that you can have all the nice soluble proteins and the oils without any of the gritty almond stuff in there. It doesn't seem like almond milk is going away anytime soon, so we're trying to improve it.
Great, thank you.
Thank you.
Great, thanks, Chris. Carrie, can I turn to you now for a question? Oh, sorry, you're on mute, Carrie. You're on mute, Carrie. Can't hear you. No, sorry. Yeah, I'm wondering if we're having a problem with your audio connection. Yeah, there might be a problem with your audio connection. Maybe what I can do is turn it to Elaine. Oh, do I have you now? No. I'll turn it to Elaine next, and then perhaps if you have a question, Carrie, you could type it in. I'll see, but I'm not sure what's happened. I think we've lost your sound. Okay, Elaine, would you like to ask a question?
Yes, that was a real nice presentation, Jessica. When you were selecting the enzyme for the enzyme-assisted aqueous extraction, what was the premise of that by which you chose the enzyme? Was it more to enhance the extraction, generate more nutritional value by breaking down the protein to make it more soluble? What was the goal, and why that enzyme versus others?
Sure. So this is first of all, food grade enzyme that we have used in previous research in our lab, specifically with almonds in other studies, and we found that this had the best rate of extraction. Something that I didn't mention here, but it did also improve digestibility of the protein as well. And I didn't see a significant impact in the digestibility with this. There was an impact, but it wasn't statistically significant. But that's the reason that we chose that one, was because we found that it had excellent extraction compared to others.
Thank you.
Thank you, Elaine. While Carrie addresses the sound issue, you did post a question in the chat. Is there a concern about bitterness with endoprotease, for example, in this approach?
Yes, excellent question. And absolutely, that was one of our very first concerns. We knew that we could get pretty great extraction with this, but bitterness, you know, with those peptides that are released with the endoprotease was certainly a high concern, which is why we're doing a sensory test. Some very preliminary tests of us in the lab. You know, I'd been working on this for about 2 years in a lab environment without actually tasting it. Finally being able to make it in a food grade environment and taste it, and we went, whew, it's okay. It's pretty tasty, surprisingly. Even surprisingly, the enzyme extracted milk we actually preferred to the aqueous, which I did not expect. But that's something that we are investigating right now. Actually, starting next week, we're building a descriptive panel to look at the acceptable levels of enzyme as far as you know, what level we can add that in before it starts to get too bitter. But surprisingly, we did not see a lot of bitterness in it, and that's what I was expecting. But we were pleasantly surprised to see that it was not that big of an impact.
Okay. Well, thank you very much, Jessica. I think we'll draw this round of questions to a close at this point in time. So I just want to remind the audience that the vote is now open so that you can go and cast your vote. And it's the equivalent of one judge's score, and it does impact the final placement. So be sure to vote for your favorite pitch. We will be back in just a moment, and I'd like to thank Jessica and our judges for this round. So thank you very much. And welcome back. We now have some time for some final Q&A and additional feedback from our judges, as well as questions from our audience. And so a reminder, please post your questions in the chat feature, and we will try to get those in during this particular, this final wrap-up section. So why don't we start with Elaine. Did you have any questions at this point in time that you last, or any general comments that you'd like to make to our presenters?
Yeah, well, first of all, as I probably expected based on yesterday's presentations, these were all excellent and very diverse in terms of topic and all well done. Everybody stuck to time. A lot of work went into it. So I really thank everybody for all that. And it was enjoyable to listen to. I could pose a question to Jessica just to follow up on my enzyme question. Have you looked at the amino acid profile before the enzyme extracted process to see whether the amino acid profile, are you losing any, since we don't want to lose any of the protein quality of what exists at the time, so.
That's a great question. That's not something we've investigated yet, simply because there were just too many conditions to do that. But since we get such great extraction with the enzymes, we're getting honestly close to 100%. I think I got 98 or 99% total protein extraction yield. That isn't a huge concern right now, but it is interesting, especially because it's an endoprotease that is random. So we can't really predict where it clips, and the functionality can be totally changed by that. But that's an interesting avenue. Once we have selected our final milk condition, or conditions with aqueous and enzyme-assisted, and scale that up and pasteurize it, that's when we plan to drill down and really investigate and see what's happening to the protein and the fats when we have the full-scale milk.
Thank you.
Great. Carrie, are you okay to ask a question at this point in time?
Yeah, can you hear me now?
Yeah.
Okay, hopefully there's not too much of an echo, but I had a question for Peineng, and kind of also maybe tying it into the presentation that came right after yours. Did you see any, I don't remember if you talked about what protein species you're seeing in your gels, but your precipitation method, did you see any changes in the protein profile between the 7S and 11S proteins, and how could you talk about how that could affect your gelation properties?
Yeah, sure. That's a good question. With the salt precipitation method that I use, I noticed that the protein profile changed, especially to the 7S to 11S. Usually, we see the 7S to 11S, the ratio is around 2:1. So vaseline, there are 3 proteins: vaseline, convaseline, and the legume. So it's we found that it's 1:1:1 of the ratio. So definitely more 7S of the protein, which is more hydrophilic. So it contributes to the more reversible and transparent gel that I got, because more hydrogen bond was established because of the more 7S was extracted.
Yeah, it makes me wonder if you tried to specifically fractionate it to 7S and 11S, how just a pure 7S gel would compare to what you're seeing with the mixture. That would be an interesting thing to play around with, I think.
Yeah, if it's pure 7S, the gelling ability will be decreased that I tested. So with a small amount of legume, the gelling ability will be increased, but didn't affect its thermo-reversibility and the transparency.
Great, thanks, Carrie. Chris, would you like to ask a question at this point in time, or provide general feedback?
Sure. I just want to echo Elaine's comments. I think all the presentations were excellent today. Makes our job very difficult. I just wanted to follow up again with Jessica regarding her research that she presented, and I may have missed it, and I apologize. I'm just curious, with the imposed conditions on roasting of the almonds, were there any changes in the fatty acid profile that you know of, or hypothesize any changes in the fatty acid profile from a nutritional perspective? Obviously, the polyunsaturates and MUFAs are a value proposition, and I'm just curious to know what changes might have occurred in those.
Sure, that's something that we haven't investigated yet. We could at a later stage, but because we've already selected the light roast, you kind of have to use at least some roasting to get that paste. Otherwise, unroasted, it just doesn't work on a very fine particle size, and the equipment just is not meant to handle unroasted. So I'm sure that there is some loss. We actually selected the roasting conditions as low temperature as we could to try to prevent acrylamide formation. That was actually one of our concerns. And based on industry standards for what is considered a light and dark roast, as well as some previous research from Dr. Alison Mitchell in our department on how to keep acrylamide levels low in almonds while still providing that level of roast. So unfortunately, I think we're kind of stuck with some degree of roasting. So there will be some fatty acid changes, but we have not investigated that yet.
Great. And just to quickly follow up on some of the hedonics that were discussed previously in terms of, and again, I may have missed it, but color. I'm just wondering, as these foods are used as alternatives to the traditional dairy products from cows, just wondering if there are any color changes.
There are. It's interesting. It's of course quite a bit darker, just by nature of the fact that there is more almond in this product, and so enzyme use and pH also we're finding is affecting the color. That's next on my list of sensory studies. Is actually doing LAB analysis of the milks. They're certainly darker, and the pH 9 milks are quite a bit darker than the pH 7. But we're doing some deeper investigation into that.
Great, thank you.
Thanks, Chris. And Matthew, would you like to ask or provide general feedback or a question to our presenters?
Yeah, thank you so much, everyone, all the presenter. Great job. Very nice presentation. I really enjoyed all three. So I have a follow-up question for Jessica and Peng. For Jessica, what is the current practice of almond meal producing? The almond meal we are having in the market, they roast it?
Typically, yes. So the almonds are roasted and ground into an extremely fine paste. They're usually blanched, de-skinned almonds, which is the same kind that we use. So it's not the same as the almond butter you would buy in a grocery store. This almond butter is ground so finely that it's sub 80 microns. I think your tongue can't detect the particle size. So it's extremely smooth, and it's typically just suspended in water. So it's about 98% water and about 2% almonds. And that's why it's so dilute. And again, if they added more of it, then it would precipitate out a solution. Of course, there are other brands that do it differently, and there are some brands that have more almond material in there, but you'll find that they actually have sedimentation on the bottom, and you have to shake them up quite a bit, and they're kind of gritty and, in my opinion, really unpleasant to drink. So that's why we were trying to make it creamy.
You also even find in your result, like increasing the surface hydrophobicity, right? You may also
yes.
Yeah, that might be one of the another wash out for you to see the solubility of your.
Yeah, it's interesting to see. We were hoping to avoid the use of gums to help stabilize the emulsion and just rely on the proteins, hence investigating the hydrophobicity and zeta potential. There is so much oil in it, though. Almonds are 50% fat, and it's good fat, but it's still hard to keep that in solution. So we are tinkering with different emulsifiers like lecithin and gums like gum arabic and gelatin gum, as well as the stage of the full-scale homogenization with the high-pressure homogenizer should help address some of those issues with the emulsion separating.
Great, thank you. So for Peng, did you measure the minimum gelling concentration of chickpea or pea versus gelatin? Like you compared gelatin versus pea, right? The minimum gelling concentration are same for these two proteins?
Yeah, for the pea protein that I extracted, in this case, the minimum gelling concentration will be 8%. Still much higher than the gelatin. So which 1% will give us a gel.
Okay. Thank you.
Great, thank you. And I'd like to take chair's prerogative and ask a couple of questions, if you don't mind. First one's to Yuran. Yuran, you've presented some very nice data on the development of the fibrils. But I'm just wondering, you were looking at proteins sort of in isolation. Do you have any evidence, or can you comment on what the potential impact would be of other materials that could interfere with fibril formation, such as starches or other products that might be present within your test solutions?
Yeah, absolutely. So in our experiment, like you said, we try to keep out of starch or other such as fat, to keep it out of our system, to make it only protein gel. But from some previous experiments, we see that the incorporation of fat or starches could definitely affect the entire system. For example, when we incubate the crude extract, which has some polysaccharides and fat in the system, after the incubation, it will form a thicker system than the 7S and 11S isolates. So that is something that might affect, that might contribute to the changes. And also the other thing that has been going on in our lab is to study the effect of polyphenols on the amyloid fiber formation. And we see that some polyphenols from other legume species, such as plantains, they might inhibit the fibrillation.
Okay, great. No, that's an important component. So I'm glad that you're investigating that. Peineng, we have a question from the audience. What can you tell us about the sensory characteristics of your gels? Have you done any sensory work on those gel components?
Yeah, unfortunately, we haven't done any like sensory analysis on this pea protein gel. Yeah, but we add like citric acid to decrease the like pH to get a transparent and thermo-reversible gel. It smells good, at least. Yeah, definitely we want to do some sensory analysis on this pea protein gel in the future.
Well, great. Well, I think we'll draw this question period to a close. I think we've exhausted the questions from their audience, and our judges have had an opportunity to ask us some follow-up questions. We've reached the end of our time. So I just want to thank all of our 3 presenters today. And in fact, all of our 7 presenters over the course of the last 2 days provided excellent presentations and bode well for the future of food science and nutrition as we move forward into the future. So I just want to congratulate all of our finalists. And we'll take a break right now. Just a reminder that if you haven't already completed the audience poll, please do so. And because the audience votes count towards the student ePoster pitch competition scores. And so again, we'll be tallying those scores and we'll be announcing the winner during the Sustainable Protein Forum, which is happening next week in Chicago, Illinois. So with that, we'll take a quick break and I'll be back with some closing remarks. Well, thank you for joining us for day 2 of the student ePoster pitch competition from AOCS. I want to thank all of our competition participants. We've had 7 excellent presentations over the last 2 days, and it's made the job for the judges extremely difficult. But we do look forward to presenting our finalists, the names of our winners next week at the AOCS Sustainable Protein Forum in Chicago. I also want to thank our audience for participating and for voting for your favorite pitch. And finally, a special thanks to our sponsors, CalSec and the Good Food Institute, who funded the competition. And so this is a very important component. So I want to recognize both CalSec and the Good Food Institute for their support for the student ePoster pitch competition. I do hope to see you at the Sustainable Protein Forum next week in Chicago. It's either in person or online, and there's still opportunity to register, and registration is still open for in-person and online attendance. So I encourage you to go to the AOCS website and look for that information. So with that, I want to thank you all, and this will draw to the close the student ePoster competitions, and we look forward to seeing you all in Chicago next week. Take care.